The first step of the flow of energy from a glucose molecule to ATP is glycolysis, and this occurs in the cytoplasm, and it doesn't require oxygen. Glucose is then converted into pyruvate, which takes a little ATP energy from itself for glycolysis to start. And the net yield is approximately 2 ATP molecules and 2 NADH molecules, which are coenzymes that has the ability to transfer electrons. The second step is the Krebs cycle, which occurs in the mitochondria, and this step requires oxygen. The pyruvate that was made is converted and will be oxidized, and this produces carbon dioxide, and we produce 2 ATP, 6 NADH, and 2 FADH2, which is also a coenzyme, and it also helps transfer electrons to make even more ATP. The third step is the electron transport chain, which also occurs in the mitochondria and also requires oxygen. The electrons are then transferred from NADH and FADH2 to several electron carriers. These are used to create a proton gradient, and the protons are used to power an enzyme called ATP synthase. This enzyme takes phosphates and adds the ADP, and it ends up making ATP, and oxygen is the final acceptor of the electrons. When the oxygen is combined with the two protons, you get H2O, and this transport chain results in 34 ATP. And with the grand total of 38 ATP from glycolysis, Krebs, and electron transport chain. The difference between the cellular respiration and fermentation is that fermentation allows glycolysis to happen with little to no oxygen, which means the fermentation is making ATP when there is no oxygen. In glycolysis, you use 2 ATP per glucose molecules, and you'll also produce 2 NADH, and now the NADH that would be delivering the electrons gained to the electron transport chain if this was aerobic cellular respiration. And once losing their electrons, NADH would be oxidized into NADH. Once losing their electrons, NADH would be oxidized into NAD plus and be involved in the electron transport chain. But since fermentation doesn't use an electron transport chain, fermentation allows NADH to give its electrons to an electron acceptor with either a pyruvate or a derivative of pyruvate. And alcoholic fermentation is done by several types of yeast. We go through glycolysis again, which results in 2 NADH, 2 net ATP, 2 pyruvates, and the 2 pyruvates that are used, which will ultimately produce carbon dioxide and 2 ethanol. And there is another electron acceptor, which is acetaldehyde, which can act as an electron transport acceptor.